In this video, we will discuss about how this adipocyte can suppress the tumor progression in the in vivo studies. And here they use immune compromised SCID mice. In the previous video, we have understood the results in the in vitro studies, that is in the cell culture studies. Here they check the impact of adipocytes on the tumor progressions in the immune compromised mice, and they use the adipocytes in the organoid form, as it will give them a 3D structures, and the results are the results replicates more closely to the biological conditions that are found in the um, human beings. Uh, they also have the options to co-transplant these adipocytes along with the xenograft implantation of the cancer cells. But what they did is they use the organoid and then inject them into the cells, as it will give a more better biological model compared to the case where both cancer cells and adipocytes are co-transplanted. So this is in the panel A, it shows the uh, how they did their experiments. So first they planted these cancer cells using xenograft. In the first they transplant the cancer cells using this xenograft method, and after 6 to 8 weeks, once the cancer is developed, they inject this CRISPR AAV adipose organoid that has this high expression of this UCP1 genes, and they used only UCP1 as they see better results compared to the other genes that they used in the in vitro studies. After 3 weeks, they checked the tumor volume growth, metabolic genes, hypoxia, angiogenesis, and first in the panel B they shows the results about the this tumor size. In the tumor size, we can see that for the control case, they see some decrease in the growth, and for these UCP1 genes expressions, they also see a reductions in the growth. And this is for the breast cancer MCF7 uh, xenograft cancer cells. And for this case of this TN, and when we quantify this breast cancer MCF7 tumor volumes, we can see that when they use the UCP1, the decrease in the tumor volume is much higher and is statistically significant compared to the control case, where they use where instead of the cancer cells they use uh, instead of this UCP1 transplanted organoids, they use different VP64 transplanted organoids, and VP64 does not cause high consumptions of the nutrients. So, and that's why they used it as a control. And for this TN breast cancer, that is MDA MB436, also they see the similar results. That is for the UCP1 genes co-transplanted organoid, they see a significantly low tumor volume compared to the control case, and similar results they have seen for PANC 10.05 and DU145 cells. Thereafter, they check different markers. First, they check the proliferation markers in terms of this Ki67. In all the 4 conditions, they can see in the immunofluorescence image a decrease in the signals for the Ki67 compared to the control. The quantifications also shows the same results, that proliferation markers goes down in all the 4 cancer cell lines that they used when it is co-transplanted, or when the adipose organoid is transplanted with this UCP1 gene expressions. UCP1 genes transfected adipose organoid is used. And when they check for the hypoxia conditions, they use these CA9 markers. We can see that the CA9 signals are also low compared to its control case in all the 4 cancer cell lines, and this also indicates that hypoxia is also low in the cancer cells that are treated with this UCP1 gene transfected adipose organoid. And lastly, they show the results for the angiogenesis or we can say that vessel density using the CD31 markers, and here also they see a low expression of the CD31, indicating that low vesicles density, and this will cause low angiogenesis formations in all the 4 cancer cell lines that are transfected with this, or that comes in contact with this CRISPR AAV adipose organoid with these UCP1 gene expressions. So all this indicates that from the in vivo studies also, the adipocytes can able to suppress the tumor progressions in vivo. Adipocytes can able to suppress tumor progressions. This is clearly evident from their in vivo results also, and there's some more studies and that are in the supplemental figures. And if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.